Thank you, Gail. Thank you very much to the organizers. I'm incredibly excited to be here, my first trip to China. Really looking forward to interacting with everyone during this meeting and thrilled to kick it off by sharing our work. The title of my talk is A Retrotransposon in Cancer, the Marker and the Mutator. I'll be focused on our work on line 1 and its expression and activities primarily in human malignancies. Now, this audience needs no orientation to the idea that much of our genome is interspersed repetitive sequences derived from the activities of mobile genetic elements. Line 1 really dominates this activity today. Line 1 elements make up 17% of our genome, and as I'll describe, it remains the protein coding retrotransposon that drives its own propagation in genomes. Also, a very significant portion of our genetic makeup are non-coding sign elements, which are mobilized by line. The ALU elements, for example, make up about 10% of our DNA and continue to be very active, particularly in the germline. I won't focus many of my remarks at all on endogenous retroviruses or the LTR elements. Suffice it to say that these are sequences derived from exogenous retroviral infection that then have stayed in our genome. And I won't speak about the non-retrotransposons, the DNA transposons, or cut and paste elements. They're really long dead as mobile genetic elements, with the exception of domesticated forms of these transposons, such as the RAG recombinase that drives VDJ recombination. So a little bit to line 1 sequence. Line encodes a bicistronic transcript, encoding 2 open reading frames and then 2 open reading frame proteins, ORF1P and ORF2P proteins. So ORF1P is a homotrimeric RNA binding complex. For the purposes of our talk today, I'll describe it as the cancer biomarker. And then where cells express open reading frame 1 protein, we view those cells as also potentially having active transcription of lines with intact open reading frame 2 protein, which really encodes the mutator functions of the element, an APE like endonuclease domain and a reverse transcriptase domain that make a nick in the host genome and reverse transcribe a copy of the line RNA that encoded it to give you a new integration back in the genome. Our understanding of this biology is that the evolutionarily young elements encode these proteins, and that each of us inherits a different complement of potentially active sequences, and that cells have to suppress line expression and mutagenesis as really an essential cell function. And forward genetic screens have intimated that DNA methyltransferases, Hush complex, and other factors are important in line silencing. I won't focus a lot in this talk on those specific mechanisms today. Suffice it to say that when one of these active line 1 loci is expressed, you can have transcription of an RNA Pol2 driven transcript to give the bicistronic intermediate of retrotransposition. The proteins then, open reading frame 1 protein and open reading frame 2 protein, as well as other proteins associate with the RNA in an RNP complex. And then the new insertion process is termed target primed reverse transcription, again with the endonuclease nicking DNA to liberate a small target site that then can base pair bind with the poly A tail of the line RNA, setting up for a priming reaction to give you a new integration. And the fact that line is active in the human genome was first really recognized by Hank Kazazian and colleagues at Johns Hopkins when they recognized line insertions recurrently having happened in an exon of a coagulation factor, the factor 8 gene, giving rise to inherited or heritable hemophilias. Haig and his colleagues wrote in that paper that insertion of line 1 elements involving the retrotransposition of a DNA sequence through an RNA intermediate and into a new and distant location in the genome represents a mechanism for mutation to produce human disease fundamentally different from those previously described.
and we completely subscribe to this really prescient comment written in the late 80s in his report. And we continue to really be interested in exactly these mechanisms today in cancer. Two areas getting a lot of attention and interest now is the potential that line reverse transcription could create cytosolic nucleic acids that prompt cellular immune phenotypes. We're also very interested, and I'll speak much more about this today, on the potential for target primed reverse transcription to go wrong and give a lesion a double-strand DNA break. So first, though, a little bit about line expression in cancers. One of the first contributions my lab really made in the field was developing some of the first monoclonal reagents that then became widely used to detect line 1 encoded proteins. And when we first had good reagents in hand for open reading frame 1 protein, we extended prior observations with polyclonal reagents that this protein is expressed in cancers and really appreciated that it's broadly expressed in a wide variety of cancers, not only restricted to germ cell tumors, for example. We can see the protein accumulate in colon cancers, and that's a pattern that we see up and down the gastroesophageal sort of tract. We see expression in esophageal cancers and gastric cancers and small bowel cancers, as well as colorectal cancers. We see expression in pancreatic cancers. We see expression in lung and head and neck cancers, both adenocarcinomas and squamous cell carcinomas of the lung. We see expression in breast, ovarian, and prostate cancers as well. Ovarian cancers actually have some of the highest levels of accumulation of this protein in high-grade serous ovarian cancers. And ovarian cancer inception is a great opportunity to highlight how in some tissues this is like a binary marker of the malignant transformation. So our thinking in the field is that high-grade serous ovarian cancers actually arise not from the ovaries, it's a bit of a misnomer, but rather from epithelial cells that line the fallopian tube. Those cells normally do not express open reading frame 1 protein, but in the so-called stick lesions, precursors of high-grade serous ovarian cancer, where p53 is mutated and the cells begin to accrue chromosomal instability, you can see very stark induction of open reading frame 1 protein expression. Okay, so at this point, we had a nice marker for malignancies in tissue. What we're really excited about and driving towards today is detection of that protein actually in non-invasive manners, in particular in blood samples from cancer patients. And we've been developing these assays in close collaboration with David Wald's laboratory. So I'll share with you today some data from Marty Taylor and Connie Wu, working closely with my group and with David's group. I should also say that this has been a really wonderful collaboration with John LaCava's lab, who has made some of our best now affinity reagents for open reading frame 1 protein. So I'll show you some of these data in the next slide, but I want to first describe the assays. One of our strategies here is the so-called Simoa assay, which is very much like a digital droplet sandwich ELISA, where open reading frame 1 protein is sandwiched between two affinity reagents, one of which captures it adherent to a small bead. Those beads are important because then you can segregate single molecules of the protein into these small wells and have the accumulation of a very strong signal for a quote-unquote on bead. And then a second affinity reagent brings in a detection enzyme. So when we take normal peripheral blood from individuals who we think are healthy controls, you can see that most people in these first generation Simoa assays do not have circulating open reading frame 1 protein in their peripheral blood. These are data from about 400 healthy control patients. You can see about 1% of individuals have a scattering of open reading frame 1 protein detectable with these ultrasensitive assays. I'll come back and tell you about one of those individuals. Suffice it to say that the landscape is quite different when we take blood samples from patients with advanced tumors of various kinds. And in particular, in women with ovarian cancers, you can see we're detecting circulating open reading frame 1 protein in a majority of those individuals. We can also detect colorectal cancer patients with circulating open reading frame 1 protein levels as well. Back to one of our control samples, we recognize in retrospect that this individual here is a gentleman who had widely metastatic prostate cancer as well as a cutaneous T-cell lymphoma diagnosed subsequent to donating this blood sample to the biobank. And so clearly some admixture of patients with cancer in our healthy controls. We don't know if there are normal circulating open reading frame 1 protein levels or if these other individuals have an occult malignancy at this point. We just don't have more information about them. So this was very exciting and encouraging. We have gone on to continue to iterate, making rabbit monoclonal antibodies and recombinant camelid nanobodies, again with John LaCava's laboratory, to continue to refine the sensitivity of these assays, to have analytically more sensitive assays and lower the lower limit of detection of these assays. You can see that many patients with cancers, this is a different cohort of ovarian cancer patients given to us by Ronnie Dropkin, the University of Pennsylvania. You can see that depending on the assay, we still are missing some of these patients, so we wanted to continue to develop better assays. This is a set of our generation 2 Simoa assays where you can see we're really, again, decreasing the lower limit of detection. At potentially the most sensitive assays, we may be detecting we're not sure if this is background or real signal, open reading frame 1 protein and controls, but still a very nice discrimination between these women and women with ovarian cancers. And in this cohort, Ronnie mixed in very small numbers of women with stage 1 and stage 2 ovarian cancers. You can see, actually surprisingly to me, that we're able to detect those individuals as well with high levels of circulating line protein. This opens up the possibility that we could use this assay for cancer detection, something that needs a lot more work now in the translational space to impact patient care. Another potential application that we've written about is monitoring patients after they've been treated for malignancies and assessing minimal residual disease states. You can see here we have a small cohort of patients with gastroesophageal cancers who were treated with neoadjuvant chemotherapy and radiation therapy and were divided through imaging studies and other clinical criteria into those individuals who responded to this treatment versus those who were primary refractory and resistant to this treatment or non-responders. You can see that there's a range of plasma open reading frame 1 protein levels at the outset pre-treatment, and then when these patients were treated, we had in first generation Simoa assays, all of those who were deemed responders dropped to undetectable levels of the protein, whereas people with persistent disease had persistent open reading frame 1 protein levels detectable in the peripheral blood. A reviewer of our paper said that's interesting. I think actually these individuals are different from the beginning, that people with higher levels might be less likely to respond, and I agree with very, very small numbers. It seems that perhaps that's a trend that's true, much more work to do on this. And we don't know at this point if these individuals are at higher risk of failing therapy because they have greater tumor burden, and that's what's being reflected by open reading frame 1 protein levels, or if there's more genetic heterogeneity, for example, or something different biologically about these cancers. So certainly more to learn. We can take gastroesophageal cancer patients as well as colorectal cancer patients and just break them into those individuals at disease presentation with high levels of open reading frame 1 protein in blood versus low. And you can see that the trend holds across as well, that individuals with relatively higher quantities of the protein in their peripheral blood tend to have a lower overall survival, although both groups really have substantial loss of life owed to this disease. So at this point, I'm going to pivot and say more about the mutator part of the line 1 element, but I want to summarize with where we are in understanding the expression of this marker. So your genome is full of transposable elements. These are not only relics and fossils, but we all have a subset of active protein coding transposable elements, line 1 elements in our genome. A small subset really are those that are protein coding sequences and driving ongoing mutagenesis. We know that the activity generates genetic variation in human populations and rarely genetic disease, like that insertion that really led Haig and his colleagues to recognize that there were active lines in humans. And our group has published more on genetic variation and potential consequences of common inherited variation in these sequences, although I won't talk more about those studies today. What I want to focus on in this discussion together is line expression in malignancies. We now know that that's commonplace across a wide variety of the most commonly occurring p53 mutated epithelial cancers in adults. Many groups before us had indicated that these cancers have hypomethylation of the genome, either broadly or hypomethylation specifically of the line 1 promoter, and so it fits very nicely that those tumors become overexpressors for open reading frame 1 protein. And in some tissues, not all, but the fallopian tube epithelial example is a great one. It really seems like a binary marker of histologic transformation. Open reading frame 1 protein now is straightforwardly detectable in tissue biopsies using antibodies from several different labs. And also, excitingly, we're detecting it in the peripheral blood. That requires ultrasensitive assays like the Simoa assays or others, the Mosaic assay being developed by David Walt's lab. So more to come as we refine its detection in the blood. I think we have a lot of work in front of us to really perfect those assays and to increase their availability for clinical and translational research. So what does it mean if a cancer expresses line 1 sequences? What's the consequence of that? So it's become clear over really several years with a boom of work starting really with Scott Devine's lab in 2010, that line 1 is doing what it was meant to do as a retrotransposon in these malignancies, causing canonical insertions that are pervasive in all of these types of cancers where we see open reading frame 1 protein expression. The activity is potentially stemming from several different line loci, and line mutagenesis can occur throughout tumor evolution. You can see here a breakdown from Jose Tubio's analysis of one of the pan-cancer genome working group studies, that there can be scores or even hundreds of somatically acquired line 1 insertion events in esophageal cancers, a range in head and neck cancers and in lung cancers, in colorectal cancers, as well as other cancers, ovarian cancer sort of falling into this category. Any time that we have driven towards better detection of these sequences with targeted sequencing strategies, for example, we can see that these are underestimates, but it underscores the activity that is present. Our lab has contributed to this literature looking more deeply at pancreatic cancer and ovarian cancer. This is a Circos plot from just one of those studies. We had patients who post-mortem had donated tissue from pancreatic ductal adenocarcinomas, and you can see these little hash marks around chromosomes show on the outer wheel instances where there were somatically acquired line insertions found in the primary tumor, and then the inner wheel marks sites that were found in a metastatic lesion in the same individual. The large arrows showing you line insertions that were shared between the primary tumor and the distant site of the disease. We think for the most part, these are non-recurrent insertions that aren't hitting special hotspots. Most we view as likely to be passenger mutations, although there are exceptions. Very clearly, about 1% of colon cancers actually have a driving mutation, a loss of one of the APC tumor suppressor gene loci by a line insertion in an exon of that gene. So again, rarely causing a clear cancer driving insertion. So work in my lab began to turn away from just cataloging new insertions in cancer genomes. And we started to ask some very fundamental questions about what might this activity mean for cancer cell growth. And these studies really began with Dan Ardelian, who then kind of refound that line overexpression can be quite genotoxic in cell systems. So here's a really striking example from one of Dan's first experiments. He had a transgene to activate line 1, a GFP control, and you can see in these particular cells, these are RPE cells with intact p53, that when he induces line expression, he completely, almost, loses clonogenicity of this cell line. The relative viability of those cell clones was really cut back. So it was clear that at least in this type of overexpression system, line was not promoting cell growth. It was doing exactly the opposite. Now, these cells have intact p53. p53 is a major tumor suppressor that really activates a specific transcriptional program. When line is induced, we can see upregulation of a number of p53 pathway targets. And p53 becomes important for inducing essentially an arrest, a lack of clonogenicity here. If you do a genome-wide forward genetic screen, you can see a huge increase in the positive selective advantage cells gain by mutating p53 when there's forced co-expression of line 1, really suggesting that, that line expression in these cancer contexts may help to promote p53 mutation. Certainly cells can't continue to grow, coping with lots of line-associated damage unless they lose p53. Depending on the cell model that you're in, if you force line overexpression, Dan also reported, and this has been corroborated by several groups, that line can also promote an interferon signaling response. In RPE cells, actually, that's one of the things that really dominates the landscape of gene induction patterns. You can see a whole host of interferon-stimulated genes, really, in some cases, massively upregulated. These are log2 fold changes graphed here. Dan also went on to do self-fitness screens in this context, basically using a TET-on luciferase as one control, a TET-on line overexpression system as another, generated pools of knockouts using CRISPR, compromising every gene in the genome, and then asked about the representation of those CRISPR guide RNAs in these two pools to find gene knockouts that had a differential effect on the growth of line-expressing cells. He found, for example, that ADAR1, which is an RNA editing enzyme that is known to become absolutely essential for cells undergoing an interferon response, in fact does become an essential gene in cells expressing line. You can see the guide RNA levels representing the growth of ADAR1 knockout cells really drop out of these cell pools uniquely in the line expressing controls, whereas the knockout is relatively well tolerated and you see the cells persist in luciferase expressing controls. Very few gene knockouts make cells expressing line grow better, but in this experimental system, one of the very best things you can do is mutate the interferon receptor, and that would be consistent with this whole pathway slowing protein translation and cell growth. Interestingly, another advantage in the absence of p53 in these cells is APC. So losing APC is especially good if you're a line-expressing cell. Now, what genes become really essential for the growth of line 1 expressing cells? So getting rid of p53 is important, and if you are working in a p53 deficient background, one of the gene families that really has a pronounced synthetic lethal interaction are the Fanconi anemia genes. So the Fanconi anemia DNA repair pathway, including core complex member Fanconi A, one of the lesion recognition proteins, FANC-M, the Fanconi anemia ligase that in turn monoubiquitinates FANC-D2 and FANC-I effector complexes, and BRCA1, which participates in Fanconi-mediated replication-coupled DNA repair. All of those genes become essential for cells to cope with line-associated mutagenesis. Dan found correspondingly that expression of line induces the formation of Fanconi anemia nuclear complexes in S-phase cells. That's graphed here. It's actually really very similar to treatment with hydroxyurea. If you compromise the line by mutating the reverse transcriptase, those foci are reduced, suggesting that a big component of line-associated DNA damage for this pathway to clear up is not just the endonuclease nicking DNA, but also has something to do with that reverse transcriptase effect. So then we've gone on to ask more questions about this and to really try to take in a global view of what line expression is doing to cells in terms of its impact genome-wide. And tackling these experiments in our lab currently is a talented fellow, Carlos Mendez-Durantes, and we're undertaking this in close collaboration with Changzong Zhang, who's an assistant professor of data sciences at the Dana-Farber, without whom we wouldn't have tackled such aggressive whole genome sequencing studies. But suffice it to say that Carlos repurposed Dan's expression system, taking this time p53 null RPE cells with a line-inducible system, has a control where here they're treated with DMSO instead of doxycycline to turn on line. We have a number of other controls reported now in his bioarchive paper. Carlos then did single cell sorts and either did a whole genome sequence analysis to understand the impact of line 1 expression in those cells in very short term, or grew out clones of those cells to then do whole genome sequencing and see the evolution of line-associated DNA damage. So what do we see? If you start with just single cells, you can see in these p53 compromised cells, sometimes there are copy number alterations in the cells at baseline in the DMSO controls. These are almost always terminal losses of the end of a chromosome associated with a DNA break that's naturally occurring. They're almost never more complex. However, when you take doxycycline and treat these cells, forcing overexpression of the line transgene, you can see that cells with 1 to 4 copy number alterations are really increased in frequency, and indeed you find a population of cells with more significant numbers of copy number alterations. Terminal losses still dominate, but now we can see gains emerging and more complex alterations in copy number as well. This is not something that the cells like, just as I showed you before with Dan Ardelian's experiments. Clonogenicity drops way off with line overexpression, but the copy number alterations do persist in surviving clones, fewer of them, very, very few in control samples, and copy number alterations persist, albeit lower numbers of them, as clones grow out, having been exposed to line 1. Interestingly, the landscape now does not only include the terminal losses, but also has this category of subclonal losses and rarely other subclonal alterations. And I'll describe to you a little bit more how those evolve as these clones grow out. So the first thing that we came to appreciate in looking more deeply at these sequencing data is that line 1 is, again, doing what it does. It's making copies of itself and also pseudogenes making pseudogene copies of mRNAs that become entangled with its reverse transcriptase. Sometimes these can be found at DNA breaks, and sometimes the DNA breaks are not sealed to give you sort of canonical surgical line insertions, but rather you have DNA breaks that then end up juxtaposing different genomic locations in bizarre ways. So here I'm showing you an example of a translocation, this one between chromosome 16 and 17. We have on one of the derivative chromosomes exactly those types of products, the recoded line copying itself, copying a pseudogene, and then these appear to be end-joined. The opposing derivative chromosome does not have that footprint of retrotransposition, but clearly we think line was involved in producing these breaks. And you end up with a balanced translocation between 16 and 17. If that doesn't interrupt a critical gene that can be well tolerated in these cells, the clone can grow out over time. What gets a little bit more interesting is when you have unstable chromosomes that are produced by this type of activity. So here's a nice example of that. This in a clone that grew out after line exposure, we found an example of a dicentric chromosome that became an unstable chromosome, associating one end of chromosome 5 with one end of chromosome 18. So here you can look at just the copy number patterns, and you can see that there's been a loss of this end of 5 and this end of 18. You can see that the two ends actually both have reverse transcribed gene products here that are joined head to head, creating the dicentric chromosome. And then there's this pattern adjacent to the loss of a subclonal or secondary loss. And this is consistent with a dicentric chromosome that was formed and then kicks off a series of breakage fusion bridge cycles where the clone grows out and you have varying points of a secondary break and some associated copy number loss in some cells in the clone. Another example of unstable chromosomes is shown here. This time not a dicentric chromosome, but an acentric derivative chromosome was formed between the end of chromosome 7, here now in very high copy number, and one of the homologs of chromosome 15, also at increased copy number, even though there's been a loss of upstream chromosome 15 sequences. We see, in addition to these two pieces coming together, actually very complex interactions and breaks and refusions within those pieces that were part of the acentric structure. And where the two chromosomes actually join together, we see a pseudogene integration. So our working model here is that line broke the end of at least one of these chromosomes, and then you have the acentric pieces that fuse together. We are presuming that this may have been segregated into a micronucleus, leading to chromothripsis and increased, complexity of DNA damage for that then unprotected part of the genome. And consistent with this, we see a real increase in the frequency of cells with micronuclei when we force line expression. So with that, my time is growing short. I want to pause and summarize some of our observations from this mutator section of my talk. It's clear from our work and many others that line is a mutagen in cancer genomes, causing numerous canonical insertions in those genomes, some of which are cancer-driving and most of which do not appear to be recurrent events, but the signature of mutagenesis is quite commonplace in these cancers. Experimental systems, if you believe them, they're not perfect cancer models, but they really do suggest when we force line expression in culture that this isn't the only consequence, that also there's a quite frequent occurrence of double-stranded breaks that can promote chromosomal instability and potentially, if this is happening in cancers, genetic heterogeneity in tumors. And some beautiful work by Jose Tubio's lab looking at cancer genomes, both as part of a published paper for PCOG as well as in a recent bioarchive, really enforce that this is a plausible model as well. Line 1 expression, we think, can promote selection for p53 mutations and prompt interferon signaling, at least, again, that's true in culture. It's unclear if these types of properties are promoting cellular transformation or malignant progression in line 1 expressing cancers. I think we need more true-to-life experimental systems to begin to rigorously evaluate this. But we are very interested in the hypothesis that epigenetic derangements at the start of cancer unleash the expression of this transposable element, and that it's contributing to the frequency of tumor suppressor mutations that we see in these diseases and karyotypic complexity that are so typical of these tumors. Whether or not line is really promoting the genome instability that underlies these cancers, I think we may be able to use line-associated DNA damage. Many of our most effective cancer therapies are DNA-damaging agents. So we're very interested in the hypothesis that cancers are doing something to limit line expression, including the expression of the downstream open reading frame 2 protein, and that we might be able to target those types of mechanisms if they exist, or further promote line expression in these malignancies, or target DNA repair pathways to really exploit our understanding of this biology. With that, I'll stop and just give a shout out to collaborators. Brad Bernstein and Alice Lee are working with us on some somatic mosaicism projects that I didn't have much of a chance to talk about today. Several other people have helped with our Lyme cancer studies. John LaCava, really key in developing affinity reagents for ORF1 studies. Marty Taylor and Connie Wu contributing to those studies. They each are now assistant professors in their own right at Brown University and at Michigan. And David Walt, really the genius behind those single molecule detection assays. Changzong Zhang, our invaluable collaborator for looking at line-associated DNA damage. And I've thanked by name several people from our Hopkins labs and our lab currently at the Dana-Farber, but Dan Ardelian's work and Carlos Mendez-Durante's work were really very much highlighted in this talk. So with that, I'll stop.
Thank you very much. So if you have questions for Katie, you have microphones on the sides, so you can line up. You need to switch it on.
Thanks a lot for the great talk. I have like a simple question about interferon response. Do you have a working hypothesis how one is inducing interferon response? Because like in some cases they don't, and others they do. What is your view on it?
This is a great question. So just to repeat, you're interested in the mechanism of line-associated interferon signaling. I think this is really still a wide open question. It is cell type dependent, although many experiments that have been done are trying to induce line expression in cancer cells, and many of those are known to have aberrant and compromised interferon responses, having altered, for example, pattern recognition receptor pathways. We're very interested in those PRRs as potential mechanisms, but what is really the driver in a different cell type, I think remains to be explored. Line induction at its own loci can produce not only a sense transcript, but also a well-characterized antisense transcript. So it's possible that double-stranded RNAs are coming from that activity, or are coming from just the prevalence of lines in the genome. If they become overexpressed, you may have line binding to line, and those double-strand RNAs could be triggers for these pathways. I'm also really interested in sequestering and DNA sensing pathways in the cytosol that could be related to line reverse transcriptase activity in the cytosol. Marty Taylor, with experiments done in our lab, have some data to that effect in a recent Nature paper describing the structure of the line reverse transcriptase. It's also possible that the DNA damage I described is sufficient to give cytosolic genomic DNA that would be triggers for those pathways. So lots of potential mechanisms, but I don't know that we really nailed it in a true-to-life cancer model yet.
Can I say something?
Maybe you can give it.
Happy to follow up with you.
Please, on the side here.
Hey, Kathleen. Thank you for the fantastic talk. And I have a question that you showed that in the ovarian cancer and colorectal cancers, the OF1P expression are the highest. So do you have any explanation why these tissues have the highest OF1P when they have cancer?
This is a great question. Long story short, we do not know. We are beginning to work on what we perceive to be normal models of both of those epithelial types to try to understand what types of perturbations produce open reading frame 1 protein, and then borrowing methods and approaches from Gael, trying to drive towards specifically what loci may be prone to be activated in those conditions. But I think more work to be done to really understand it. Great question.
Thank you.
One just comment I'll make as an aside is that this is not universal to all cancers. So CNS tumors like glioblastomas, many hematolymphoid malignancies don't have this very high degree of overexpression, and we don't have a good understanding for why line may be better regulated in those malignancies either.
Expecting for the solution.
Thank you. Johan, you want to?
Yeah, thank you very much for the talk. I had a slightly related question. I was wondering if you had looked at, if you know which loci the ORF1P signal comes from, if it's the same loci in different patients and in different tumors, or if it's extremely random?
It's a great question. We are just beginning to drive towards this now. I don't think that we have a great understanding. Some RNA studies, and we want to complement them with histone marks, with methylation marks, do show recurrent activation of some loci. And actually, a great example of that is a line that is embedded in the TTC28 locus, which is known not only to be overexpressed in colon cancers, but is one of the elements that's prone to 3' transductions, and it's known to serially mutate colorectal cancer genomes. So that's one of these hot loci, but I think we have some work to do to really figure out the complement in every tissues and address your question. We're also very interested in how much genetic variance in human populations contribute to that expression. It may be that normal open reading frame 1 protein levels in circulation or our ability to detect particular cancers will depend on the lines that someone inherits and how effectively they're silenced. So more work to be done there.
Hi, Kathleen. Very interesting stories. So I have two quick questions. The first one is, did you guys detect allele of 2P in biopsy?
Great question. Short answer is no. We have a mobile DNA paper we worked on with John LaCava where we went to great pains to develop not 1, but 5 wonderful monoclonal antibodies against the protein. We're very good at detecting the recombinant protein. We're very skeptical of readily detectable protein levels, ORF2 protein levels in cancers, although clearly there's genetic evidence that some of it is being made.
Okay. And my second question is, actually my knowledge was that when line 1 becomes active, it expresses ORF1P, then it binds to its own RNA, then goes to the nucleus for transposition, right? So I'm wondering how this ORF1P exported or translocated from the cells to the biopsy. What do they do?
Great question. Your understanding is exactly mine. We are inferring, but we don't really know, that cancer cells are dying and that's how we're getting this protein leached out into the peripheral blood. We have no evidence that it's actively secreted or that it's very efficiently accumulated in extracellular vesicles, but those are some other ideas that we've been kicking around.
Thank you. Thank you. Maybe go ahead.
I have a similar question to this. So actually, it's a stoichiometrical problem. So basically, if the messenger is translated right into the ORF1 protein, ORF2 protein, it binds to its own RNA, does it inhibit its own translation? Or it's very dynamic, can be released or dying and so on. Yeah.
So I'll say one thing about translation of ORF2, and then the best person in the room probably to speak with more about it is John Moran. His group published a beautiful study some time ago called the unconventional sort of translation of that protein using retrotransposition as a readout. John did many beautiful studies to modify that RNA and came to the conclusion that there does not appear to be an IRES, but rather a ribosome finishes with open reading frame 1 protein, rarely reinitiates translation after traversing the inter-ORF spacer. We don't know at this point what cellular processes, if any, may specifically regulate that. But we do think our failure to detect ORF2 is a production problem, more so than it being actively, rapidly cleared in cells, though we haven't totally ruled out that possibility.
Thank you. So secondly, overexpressing
Maybe we give the speech to the next
Happy to
speak.
discuss.
Yeah, and but you can stay, and if we have enough time.
By the way, this guy is our sponsor for Free Bar. Let him finish the question. So I have a very simple question. Since in your of1 expression, the detection of that, you first find a pseudo-normal, but later also proved to be a cancer patient. So my question is that how early you can detect in normal or in cancer patients for using of1 expression?
Great question. That's something we really need to figure out, right? How reliably can we detect it in early stage disease? Is there a normal baseline that's ever acceptable? How good are we at differentiating between those? We really don't know. In tissue samples, it's clear, for example, in stick lesions, we can see induction right as cancer begins, but whether we'll reliably be able to detect stage 1 disease and distinguish it from any normal sort of background levels becomes really very, very important. So that's something we have to figure out. Another thing that may be important to consider is how stable someone's level is. If we ever have assays that make normal levels of ORF1P detectable and measurable, is it something we can follow over time? Does each person have a baseline? Does an upward trend indicate that you're at a risk for then later a clinically apparent malignancy? Lots to do.
We have time for last quick question.
Yeah, sure. I will make it quick. Really great talk, Kathy, as always. Fabulous work. I have a quick question on the whole genome sequencing from overexpressing line 1 in the RFP cell lines. Do you see a lot of chromosome instability increased, but do you see any like specific line 1 mediated, line 1 retrotransposition versus like sign or other parasites of line 1 being retrotransposed that is causing this chromosome instability?
So we do see canonical line insertions. I really did not emphasize that at all in describing this. You can look in the bioRxiv paper to see more of those. We see lots of line insertions, not so much in the way of ALU. We do see a lot of pseudogene insertions, which may be an aberrant thing in our system, or maybe we're under ascertaining those in cancer genomes. Our thinking now is that almost for every one of those canonical integration events, we're getting a DNA double-stranded break that's not repaired appropriately. Whether that ratio really holds as we move to more true-to-life models, expressing, for example, endogenous elements at the levels of expression that are really seen in cancers remains to be seen. If that's true, if a line insertion happens half the time and a break at that locus happens half the time, that could really imply that there are profound contributions of line mutagenesis to chromosomal instability in cancer. So we're scratching our head about that and very interested to know if it might be true. Thanks.
Thank you so much. Congratulations.